Right now, as you sit here, a sprawling, hidden city is constantly at work inside you. If we push past the surface, through the dense matrix of your skeletal system, we find the dark, spongy center, the red bone marrow. This marrow operates as a ceaseless, silent assembly line. Every single second, it manufactures millions of new cells to keep your body functioning and alive. But before these newborn cells become specialized tools, like oxygen-carrying red blood cells or pathogen-hunting immune cells, they exist in a before state. They are stem cells, raw, undifferentiated biological blank slates. Without these hidden master builders constantly churning out fresh material, your physical body simply could not survive the daily wear and tear of existing. There is a strict biological rule at play here. Every single specialized cell in your body contains the exact same instruction manual. They all carry the identical DNA blueprint. This creates a biological paradox. If the manual is identical across the board, how does a blank slate stem cell actually know whether to turn into a rigid piece of bone or a fluid red blood cell? A mature cell is locked into its role, characterized by rigid permanence. A stem cell, however, is defined entirely by fluid potential. It just sits there, waiting for instructions. Deciphering how these cells navigate their choices reveals the mechanics of how the entire human architecture is built from a single instruction set. To see where this choice begins, we must rewind to the earliest stage of human development, where cells operate at their absolute maximum level of potential. In this diagram, the lower left shows fertilization forming a diploid zygote. This totipotent egg initiates human life. Following the arrow for mitosis, it divides into pluripotent embryonic stem cells, having the freedom to become any specific cell type. Early cells are vastly different from the adult stem cells currently working in your bone marrow. Those adult cells are multipotent. Their pathways are restricted, meaning a blood stem cell can only become a blood component and nothing else. Biological development requires a strict trade-off. To gain a highly specialized, necessary function, a cell has to sacrifice its infinite potential. If we drop back down into the localized environment of your marrow, we can observe the precise mechanism that forces a cell down one of these restricted paths. The physical transformation of a blank slate into a specialized worker is called differentiation. It's driven by chemical cues from the environment and the cell's own internal genetic instructions. Think of environmental cues as switches. The cell reacts by turning specific genes on and others off. By reading only these illuminated sections, it alters its internal structure, morphing into a specialized worker like a red blood cell. Differentiation is the engine of biological growth. It takes a chaotic mass of pure potential and turns it into a highly organized, functioning machine. Today, this process is no longer confined to the natural environment of your bones. In modern clinical laboratories, scientists are actively working to hijack this biological engine. This is the core of regenerative medicine. Doctors are figuring out how to harvest these master builders to repair failing organs and replace dead tissue. For a patient battling leukemia, their marrow assembly line is producing defective immune cells. By transplanting healthy stem cells, doctors can physically override and replace that malfunctioning factory with a healthy one. Regenerative medicine moves us past managing the symptoms of a disease. It allows doctors to actually regrow the body's hardware. Harnessing that power comes with societal friction. Attempting to harvest early-stage, highly flexible embryonic stem cells sparks intense ethical debates about human life and medical boundaries. To navigate this, the scientific community relies on the ISSCR guidelines, a strict international framework designed to ensure ethical oversight and the responsible clinical use of this science. Meanwhile, the research continuously adapts. Scientists are seeking new methods to unlock cellular potential, like reprogramming adult cells to act like embryonic ones, bypassing the need for controversial sources entirely. These stem cells are the literal architects of human life and the active repair crew deep in our bones. Understanding their behavior gives us a path to not just treating a failing body, but rebuilding it.